Okay, from the previous exercise, we used the different algorithm to actually identify the classifier, and since the random tree give the best fit, basically is 100% match with the sample set. So we are going to use the classifier that is revealed from the random tree to do the pathway analysis. So for the random tree, there are altogether 177 classifier that's being identified. Now, in this exercise, what we are going to do is that we are going to look at these 177 classifiers, see where which pathway that they are being enriched for. That will give you some sense of what are the biological mechanism or the pathway that that might govern the response of the gemcitabine. So the first we will do is basically we use a string. So basically just to look, is a very nice, okay, a network analysis. Basically you just copy and paste the list of the genes here, over here, and then you select the organism, basically Homo sapiens. So and then you press the search button, so then it will automatically match it to the database. So then you just click continue to it, and then here you can see that, okay, how the relationship of each of the gene that or the classifiers that you identify related to each other. The one that's connected with the line basically represent that they are somewhat connected. Okay, for example, okay, this S1PR4 is somewhat connected to, okay, Amica 1 and so forth. So if you want to look at the legend, basically, so the black color line means that they are co-expressed, and then the red color is gene fusion and so forth. There's a lot of legend here basically you can look at. So this is good. So you can then from here then you can identify certain node that maybe well this group of, for example, this cluster of genes, they might play a similar role. So and that is basically being identified to be the predictors of the gemcitabine and so forth. So there's one way to look at it. However, it shows you the close proximity of all the different genes entry that you are being identified, but it doesn't actually tell you which pathway that is being involved. So to do that, you can go into another way to look at it. Basically, you can go by, for example, look at it by the gene ontology. So you can look at that, or what you can do is you can also look at clustering, or there is another, oh, sorry, you, oh, so you can also go into analysis here. So functional enrichment of network. So okay, you can also look at the CAG pathway. So for example, okay, the genes that are involved in the glycine serine